Let's take a look at the sliding filament model of contraction in a diagram. What you're looking at is a cyclical diagram showing this process in action, this four-step process that involves two molecules of ATP being hydrolyzed or split apart with every sliding motion in the sarcomere. What you're looking at in brown represents a thick filament. They have taken off all of the myosin heads except for this one pair right here. Up here in yellow, this represents a thin filament. The black line right here represents the tropomyosin. And the blue structure, this much smaller blue and pink structure right here, that represents the troponin. So we said the first thing that happens is the myosin head sort of cocks backwards. If you think about it as sort of being oriented perpendicular to the rest of the thick filament, the first thing that happens is an ATP binds to the myosin head and causes it to sort of cock backwards the way you would cock back a gun before you fired it. The binding of ATP to this protein is going to cause this protein to change shape. And the shape change is a cocking backwards. Now, remember, these heads have the ability to perform catalysis. They act like an enzyme, and they're going to break this ATP. And that's what you see in number 1 happening. The myosin head is going to break the ATP molecule into ADP and a phosphate group. So some energy has been released now. And what's going to happen is that energy is going to be used to move that myosin head up into contact with the thin filament. It's going to bind to the thin filament, and then it's going to swing forward. All right? That's called the power stroke. It's going to push, essentially. It's going to push that filament. It's going to slide that filament forward. That's called the power stroke. Now, at the end of the power stroke, we have a myosin head bound to a thin filament, but all of the energy has been used up. The sliding is finished. The only way to get this head to release is to add another ATP in. Another ATP is going to come and bind to the head, and that's going to allow it to relax and cock backwards. It's going to let go, and it's going to cock backwards. The ATP gets hydrolyzed. The energy gets used so that the head binds to the filament. The power stroke occurs. The filament slides forward towards the center of the sarcomere. Another ATP binds. That causes the head to let go of the thin filament, cocks backwards. And on and on and on it goes, as long as there's ATP available. Now you can imagine that there's tight regulation of muscle contraction. There's tight regulation of most processes that occur in the body. You wouldn't want muscles contracting unless there was a reason for them to contract, unless the central nervous system wanted them to contract. And so contraction is regulated, and it's regulated using calcium, using free calcium ions. Those ions are the molecule that's going to help control this process. Remember that the sarcoplasmic reticulum inside of each muscle fiber stores calcium and can release it very rapidly along the entire length of the muscle fiber in those triad systems. So what happens when the sarcoplasmic reticulum releases all that calcium? Well, the calcium is going to flood over all of those myofibrils that are inside the muscle fiber. And that calcium is going to bind to that little troponin protein that sits on top of all of those thin filaments. It's going to bind to that TNC region of troponin. Troponin is just a protein, and anytime something binds to a protein, it's going to cause it to change shape. The TNC part of troponin is going to change shape, and that shape change is literally going to push the tropomyosin molecule, the licorice stick molecule, out of the way, away from the groove. And that's going to allow the myosin heads to bind to the thin filaments. As long as there's ATP available, once the tropomyosin is moved away from the groove in the thin filament, the binding sites are exposed, and now the myosin heads can bind, and cross bridges can form. Calcium is the regulatory molecule. Now, this is a strange looking diagram, but let's walk through it. What you're looking at here on the left in brown, this is one thick filament that has been cut in the transverse plane. So we're looking at a cross section. We've cut through, like a slice of bread, through a thick filament. Remember, all a thick filament is a whole bunch of myosins, all bundled together. So normally we would expect to see heads jutting off of this thing in 360 degrees, but they've removed all of them except one. So we see one myosin head sticking off of this thick filament. Now up here, this is a thin filament, again in cross section. All a thin filament is two strands of actin molecules. These two strands are just wound around each other. So we've cut through that thin filament like a slice of bread. We're looking at the cross section. We're looking at the face of that cross section. One strand, two strands. There's a tropomyosin, there's a licorice stick protein lying linearly right along in the same direction, parallel to the thin filament. That's what you're looking at here in green. And then there's this other protein called troponin that is sitting nearby. Troponin has three parts. It has a TNI part, a TNC part, and a TNT part. Remember, the TNT part binds to tropomyosin. That's what the T is for. The TNI part binds to actin. The TNC is what binds to calcium. Now, the last thing to notice is there's this little black cross-hatching right here on this region of the thin filament. This is the spot where myosins can bind, where myosin heads can bind. And normally, tropomyosin is in the way. Remember, we said the binding site was near the groove between the two strands of actin, and the tropomyosin just blocks it normally. So, in a relaxed state, where there's low calcium in the area, in that relaxed state, tropomyosin blocks the myosin binding site, and the heads can't bind here. Even if there's ATP around, they can't bind here. Their binding site is blocked. Now imagine that a nerve impulse has arrived and is signaling to the sarcoplasmic reticulum to release calcium. So now we have flooded this muscle fiber with calcium. Calcium is going to bind to, here's the calcium, it's going to bind to the gold part of troponin, to the TNC part. And that binding is going to cause this protein to change shape. And as it shifts in its shape, it's pulling or pushing, depending on how you want to think about it. It's having an effect on the tropomyosin that it's attached to. So the TNC changes shape, the whole troponin changes shape, and it removes the tropomyosin, the licorice stick, away from the binding site. Now the heads can bind as long as there's ATP around. So calcium is the regulatory molecule, and it's the nervous system that is in control of when that calcium gets released. The sarcoplasmic reticulum is the place where the calcium is stored and the place where the calcium is released. And that storage and that release is in control of the nervous system. Not only can the sarcoplasmic reticulum release calcium, but it can take that calcium back inside. It can store it again when it gets the right signal. So we need a communication point between the nervous system and the muscle fiber. We need a synapse. All a synapse is a communication point between a neuron, a cell of the nervous system, and whatever cell that neuron is communicating with. Sometimes neurons talk to other neurons. Sometimes neurons talk to effector cells like muscles and glands. So where a neuron communicates with a muscle, that's a synapse, and it's a special kind of synapse called a neuromuscular junction. It's a special kind of synapse that releases a special kind of neurotransmitter. That's what gets released into synapses. Neurotransmitters, they're just chemicals. They're released out of the neuron. They travel across the space that is the synapse, and they bind to the membrane of the next cell, whether it's a neuron or a muscle cell, muscle fiber, or a gland. When calcium gets released from the sarcoplasmic reticulum, contraction happens. When calcium gets taken back up again, and that's achieved with pump proteins, relaxation occurs. And it all happens at the neuromuscular junction. The axon of the incoming nerve, really shouldn't call it a nerve, I should refer to it as a singular neuron. Because as we'll talk about later in the course, nerves are actually made up of many, many axons. But if we think about one neuron right now, one nerve cell, it's got a long axon extending from it. And where that axon comes in communication, synapses, with the muscle fiber, it's right at the sarcolemma. That synapse is called the neuromuscular junction. So in order for a signal to come down an axon, a nerve signal to come down an axon to the neuromuscular junction, the brain has to send that impulse. The brain or the spinal cord, the central nervous system has to send that impulse down along those motor neurons, the effector side. The impulse is going to reach the neuromuscular junction. And as long as it's a strong enough impulse, and again, we'll talk about threshold level when we talk about the nervous system, as long as it's a strong enough impulse, when the nerve impulse gets down the axon to the end, a neurotransmitter is going to be released. And that neurotransmitter at the neuromuscular junction is called acetylcholine. Acetylcholine is just a chemical. It's a molecule. And we call it a neurotransmitter because it's transmitting some information from the nervous system. And that information is it's time to contract. So the acetylcholine travels through that space between the axon and the sarcolemma. It travels across that space and it binds to receptors on the sarcolemma. And that causes the receptor to change shape, and the end result is that the nerve impulse will be regenerated in the sarcolemma. Remember, the sarcolemma is a special form of a plasma membrane. It is capable of transmitting electrical current. So now we have a nerve impulse traveling through the sarcolemma. Remember, we said nerve impulses travel in the form of what's called an action potential. That action potential is spreading out across the sarcolemma. It's going to be directed, though. It's going to be directed down those T-tubules. Remember those? And that way, the nerve impulse will travel through the entire length of the muscle fiber across all surfaces very rapidly. Those T-tubules are part of a triad. They come in contact with the terminal cisternae of the sarcoplasmic reticulum, and that nerve impulse is going to stimulate calcium release. So again, the brain, or again, the spinal cord could also do this, is going to send the impulse. The impulse is going to arrive at the neuromuscular junction. Acetylcholine is going to be released from that nerve ending. It's going to cross over the synapse. It's going to bind to receptors on the sarcolemma. It's going to cause the action potential to be regenerated in the sarcolemma. And that's going to travel down those T-tubules, contact the terminal cisternae, and calcium is going to get released. That muscle fiber is going to be flooded in calcium ions. You can't see the neuromuscular junction here, but here's that sarcolemma. And acetylcholine is going to cause the sarcolemma to, the word we use is depolarize. And that's going to regenerate a nerve impulse, essentially, action potentials in the sarcolemma, in the membrane around that muscle cell. That nerve impulse is going to be directed down into the T-tubules, all of the T-tubules at the same time, and it's going to cause the nearby terminal cisternae to release calcium. Now, what does the calcium do? Remember, the calcium binds to troponin C, causes it to change shape, causes it to push tropomyosin out of the way. Now the myosin binding sites are available. And as long as there's ATP around, contraction is going to begin. The ATP is going to bind to the myosin heads. They're going to cock backwards. The ATP is going to get hydrolyzed. The myosin heads are going to bind to the thin filaments. The power stroke is going to occur. Sliding happens. The sarcomere gets a little shorter. Another ATP is going to bind. It's going to get hydrolyzed. The myosin head's going to let go. And then the whole thing's going to repeat. And the sarcomere is going to get shorter and shorter and shorter. Muscle contraction. On this slide, you're looking at a different view of a muscle cell, a muscle fiber. Here are the individual myofibrils. So we've cut this muscle fiber at a couple of places. So we're just looking at a little piece of it. The blue that's sticking out here, that represents sarcoplasmic reticulum. Remember, that travels all around and in between all of these myofibrils. The yellow represents a strip of the sarcolemma. Again, we're just looking at a little short slice of this muscle fiber. And this represents an axon, the long linear extension that comes off of a neuron and carries action potentials. You'll notice that at the end of the axon, it branches out Again, we'll talk about this more when we talk about the nervous system. But this structure, this axon and its terminal branches, it needs to communicate with this membrane. So it comes down and it has these communication points, these synapses as we call them. But we give it a special name. We call it the neuromuscular junction here. It's a type of synapse that when it gets the signal is going to release acetylcholine. And the acetylcholine is going to bind to the membrane of the muscle fiber. It's going to bind to receptors and it's going to regenerate another red arrow, another nerve impulse over here. It's going to regenerate action potentials over here. Now that regenerated signal needs to spread all through this cell really quickly. So it's going to be directed down these T-tubules. That's what they're showing us here. They've removed a little piece of the membrane now so we can watch the regenerated red arrows sort of dive into the T-tubules. Remember, each T-tubule is part of a triad. It sits next door to two terminal cisternae. And if we blow up a region, we can see that triad right here. Here's a T-tubule in yellow. Here's a terminal cisterna. Here's a terminal cisterna in blue. And the signal that's coming down that T-tubule, the action potentials that are coming down that T-tubule, is going to trigger calcium to be released from the sarcoplasmic reticulum. You'll notice that that T-tubule, that triad, lies right over a Z-line. That's not super important to remember, but it just shows you that these triads are positioned every so often so that each sarcomere gets flooded with calcium at about the same time. The calcium is going to bind to troponin, and that's going to move the tropomyosin out of the way and allow the myosin heads to bind so that contraction can occur. Now, there's one other structure that I haven't mentioned yet that I want us to be familiar with. It's a structure that, or a term, that is often confused with neuromuscular junction. And that term is motor end plate. Remember, we said the neuromuscular junction is a specialized synapse. And a synapse is the communication point between a neuron and another cell. So the point where the branch of that axon, the final branch of that axon, comes in contact with the sarcolemma, that synapse is called the neuromuscular junction. But on the sarcolemma, that is a specialized area that can respond to acetylcholine because it's got acetylcholine receptors in it. So the region of the sarcolemma that is part of the neuromuscular junction, the region of the sarcolemma is called the motor end plate. A motor nerve is going to come and communicate with it, and acetylcholine is going to be released, and the acetylcholine can bind to that motor end plate because there are receptors for it there. So the motor end plate is the region of the sarcolemma. The synapse is between the neuron and the motor end plate. The synapse is called the neuromuscular junction. Those two terms are really easy to confuse with each other. So it turns out that acetylcholine is a really abundant neurotransmitter. It's used throughout all of this muscle, so it has to be quite abundant in the nervous system. It's called an excitatory neurotransmitter, and all that excitatory term means is that the action potential that comes down the neuron will be regenerated in that second cell, in that muscle fiber. It's going to excite, or it's going to regenerate action potentials in that second cell, that muscle fiber. Acetylcholine gets released from little vesicles that are stored in the ends of the axon, those enlarged sort of knobs that we find at the end of axons. Remember, the axons branch out at their very end. We call those terminal branches. And at the end of each branch, there's a little enlarged area called a knob. And that's where the acetylcholine is stored. It's stored in little membrane-bound organelles called vesicles. In order to release the acetylcholine, those vesicles have to come and fuse with the membrane and release the acetylcholine into the space, into that synapse. They do it through a process that we see in lots of cells, a process called exocytosis. Exo means out. So something's being released out in exocytosis, and that something is acetylcholine. It travels across the space between the neuron and the muscle fiber, and it binds to receptors on that muscle fiber, the postsynaptic cell, right in the area called the motor end plate. Now, that binding of acetylcholine to its receptor on the muscle fiber is going to regenerate the action potentials. And the way it does that is by opening up a special kind of ion channel called a sodium channel. And by opening up the sodium channel, sodium is going to rush into the muscle fiber, and it's going to cause a specialized state that we refer to as depolarization. We're going to talk more about depolarization and sodium channels when we talk about the nervous system. But for now, understand that when the sarcolemma gets depolarized, that is going to generate new action potentials in the sarcolemma. And it's those action potentials that are going to travel down the T-tubules and elicit a muscle contraction. Acetylcholine is a powerful molecule, right? Acetylcholine is needed to initiate muscle contraction, and you can imagine that the body has to keep tight control over how long the acetylcholine lasts in that synapse at the neuromuscular junction. The way that the body stops the action of acetylcholine is by using an enzyme. There's an enzyme called acetylcholine esterase. And this is an enzyme that breaks down acetylcholine. So the acetylcholine gets released into the neuromuscular junction, and it binds and has its actions, but it's not allowed to act for very long. It gets broken down by acetylcholine esterase. And that way, muscle contraction doesn't go out of control. If muscle contraction went out of control, the individual would be in trouble. If muscle was allowed to contract and contract and contract and contract without relaxation, the person would be in trouble. And one of the places that the person would be in the most trouble is in the muscles that surround the thoracic cavity. If those muscles were allowed to contract and contract and contract without relaxation, the person would suffocate. Scientists have been studying this important enzyme, acetylcholine esterase, for many, many years. There was a whole lot of interest in acetylcholine esterase when certain types of pesticides were being developed. You may have heard of these pesticides. They're called organophosphates. Organophosphates have been in use for decades, not just here, but around the world. And organophosphates are acetylcholinesterase inhibitors. So, an organophosphate pesticide will act upon small animals that are pests to us. Think about rodents for a minute. If a rodent eats an organophosphate chemical, an organophosphate pesticide, it's going to inhibit the action of the acetylcholinesterase enzyme, and it's going to allow acetylcholine to work well past the amount of time it was intended to work. It's going to put that rodent into a continuous state of muscle contraction. And the rodent's going to suffocate. Now, we use organophosphates against not just rodents, but any kind of small pest, any kind of an animal that's considered a small pest that has this enzyme will be killed with these organophosphate chemicals. And of course, human beings can also be killed by them. Every year there are cases of, for example, migrant workers who are applying organophosphates to crops, trying to control pests, and they accidentally get exposed to a high level of these compounds and suffocate and die. There's been a lot of activity in the regulatory community trying to get rid of these chemicals because they're dangerous, but they're also very effective and they're not expensive, which is why they're very popular on the market. Now, in addition to being used in a commercial sense, organophosphates have also been used as nerve agents. So the kinds of weapons of war that we refer to as nerve gases. There was just a case in the news recently of an enemy of Vladimir Putin named Novotny, Mr. Novotny. He was a political rival. He is a political rival of Mr. Putin's, and he was speaking out against Mr. Putin. And one day, as he was preparing to leave the country, this just happened a few weeks ago, someone poisoned a cup of tea that he was going to have at the airport with one of these nerve agents, one of these acetylcholinesterase inhibitor agents. The agent is outlawed now across the globe. It's called Novalchek, and it's a very, very powerful organophosphate. So very small amounts of it will poison a human being by inhibiting all of the acetylcholinesterase in their body and putting them into a state of muscle contraction, and they'll suffocate and die. Now, Mr. Novotny was lucky because he was taken to a hospital and he was treated. He was treated for this poisoning. He was not expected to live, but he did. He was transferred to Germany, to a hospital in Germany, and they saved his life. So he's very lucky because these nerve agents are very powerful, and very small amounts of them can suffocate and kill a human being. So let's review what we've talked about so far. We said that nerve impulses arrive through neurons to the neuromuscular junction, to this specialized synapse between the nervous system and the muscles. When the nerve impulse reaches the end of the axon, it triggers an exocytosis process. It triggers the release of acetylcholine, which travels across the synapse, the space of the neuromuscular junction, and binds to receptors on the sarcolemma, the motor end plate. And those receptors, once they're bound with acetylcholine, cause sodium channels to open, and sodium rushes into the muscle cell and depolarizes it. It regenerates action potentials in the sarcolemma, and those action potentials travel down the T-tubules, causing calcium to be released from the terminal cisterna. That calcium then floods the whole muscle fiber. It binds to the TNC of troponin, causes it to change shape, and the troponin pulls on the tropomyosin and moves it out of the way and exposes the myosin binding sites. Now, as long as ATP is around, the myosin can now bind to those binding sites. Now we're ready for contraction. The myosin has ATPase enzyme activity. The globular heads are catalytic. They can break apart ATP, and that's just what they do. When the ATP binds to the head cocks backwards. And then we see that ATP gets split. I've got them written here in reverse order, but this is all happening basically simultaneously. The ATP binds, the head cocks backwards, and then the ATP gets split. The energy from the ATP allows the myosin head to bind to the binding site on the thin filament, and the power stroke occurs. The myosin physically slides the thin filaments towards the center of the sarcomere. More ATP binds to the myosin head, and it's allowed to release from its binding site. The myosin ATPase splits that ATP, and now we're ready to go again. The ATP that binds to the myosin head allows it to cock backwards. The ATP gets split. The energy is used to allow the myosin head to bind to the thin filament, and the power stroke occurs. Another ATP binds. The myosin head releases. On and on and on it goes. The sarcomeres shorten. The myofibrils shorten. The muscle fibers shorten. The fascicles shorten. The muscle itself contracts. Now we're ready for it to end. We're ready for it to end. The nerve impulse drops back below threshold in that neuron. No more acetylcholine is going to get released now. And the acetylcholine that is in the synapse, it's going to be broken down by acetylcholinesterase. Since there's no more depolarization in the sarcolemma, there's no more action potentials in the T-tubules, the sarcoplasmic reticulum is going to pump calcium back into the terminal cisternae. That means calcium will release the TNC. The TNC will release the tropomyosin. The tropomyosin will return to that groove area, and it will cover up the binding sites. And the muscle will relax. Contraction will end. I have put together an additional handout for you, which outlines all of the steps that I have just gone through in a written form. So if you find that helpful while you're studying this very complex process, if you find that helpful, that handout is available to you on Canvas.